Hey guys, this is the process of transcription. And transcription is the first part of the process of molecular central dogma. Molecular central dogma is the idea about how information is passed along in our cells. So, the molecular central dogma consists of it's basically how DNA goes to RNA and then RNA is made into a protein. And the first part of this process is DNA, which is made into an RNA, and this process is called transcription, which is what we're going to go over. So, it starts off in the nucleus, okay? It starts off where we have our two DNA strands, okay? We have this top DNA strand and this bottom DNA strand, okay? And you can see the complementary bases like T goes to A, A goes to T, G goes to C, all of that. So this orange and orange is our DNA strand, okay? And so what is going to happen is that basically genes, we know what genes are, that they code for a specific protein, and then that protein codes for a specific trait. So depending on what protein is asked, it's only going to transcribe that gene. So basically, transcription is the copying of DNA onto RNA, that's what transcription is. So how it works is that there's two things that are needed. So RNA polymerase is the one enzyme that is needed to for transcription, and transcription only occurs on a fragment of a gene. So this is RNA polymerase, or this little red looking thingy. This is RNA polymerase. RNA polymerase in eukaryotic cells, it needs two things to know where to build. Where am I building the RNA? It needs a promoter, which is the promoter is this little greenish yellow part. It's a sequence of DNA that comes just before the gene starts, and our RNA polymerase can only bind to the promoter. And then it also needs a transcription factor. Transcription factors are proteins that hover over the promoter, like these blue dotted lines, and they show and they highlight where the RNA polymerase needs to bind onto the promoter in order to start building. So RNA polymerase, it unzips and builds at the same time. And it goes from, it reads up and it unzips, it reads up. So it's and then it writes down, obviously. So the RNA polymerase, it's going in this direction because it's reading up. It's unzipping the bonds from 3' to 5'. But then when it builds the RNA, it's going to build from 5' to 3'. And then it starts bringing the complementary bases as an RNA. So in RNA, we know that A binds with U, and then C binds with G, right? So A doesn't bind with T because that's not, thymine is not ever found in RNA. It's always uracil that's found in RNA. So it's gonna do where now our RNA polymerase is building. So now we start off at the 5'. It's this yellow strand is a new strand of mRNA, or messenger RNA. And so then T is, because A is a complementary base to T, then it makes an A. These are complementary and these are binding. U to A, G to C, C to G, and etc. But if you look closely, the RNA right here is actually copying the top strand, correct? With the exception of the uracil and the thymine. But if you look at it, A goes to A, T goes to U, G, G, C, C, G, G. It's using the bottom strand, which is this bottom strand, but it's actually copying the DNA of the top strand. And this is going to be important, especially in our translation process later on. So RNA polymerase is going to keep on, and if RNA polymerase was using the top strand and copying the bottom strand, then it would also go in this direction because, again, it's reading up, it's unzipping up. Anyways, so then the RNA polymerase is going to build and build and build until it meets the termination site. The termination site is basically just, it's in the nucleus where it's a DNA sequence, and then once it reaches a certain DNA sequence, it's going to detach itself and this RNA strand is going to fall off, And then because the whole reason is that we want to carry those genes to make proteins out and create the proteins that send for the traits outside of the nucleus. But DNA can never leave the nucleus, right? So how are they going to send that information? By making RNA. So then the RNA, but this is known as pre-mRNA, and we'll find out later, is then detached and then sent through the nuclear pores outside of the nucleus to meet with ribosomes, where the ribosomes will then carry it on to then synthesize proteins in the translation step in the cytoplasm. We know this. So now we have a pre-mRNA. So then we need to, obviously we can't just automatically go synthesize this into protein because it can maybe have some errors in it, right? So basically, then something called RNA splicing or processing is gonna occur. RNA splicing is basically where we have our mRNA, which was this yellow line, right? And then what's going to happen is that it's going to get added onto, because the pre-mRNA, it's a rough draft. It needs to just get finalized and edited, just how like when you write essays, we have a rough draft, but then we need to fix it and make a final draft, right? So there are two things added on to the pre-mRNA for RNA splicing. You have the G cap at the beginning and then the poly A tail at the end. And these are just adenines, like the nitrogenous bases. So why are these just G cap and poly A tail added in the first place? Well, it has many like things, but one, it prevents degradation from enzymes when leaving, because we'll see that we'll see in a bit that when the introns are leaving the thing, the G cap helps it prevent degradation, or helps it like not dissolve and like get bad. The G cap also helps it to navigate to the ribosome, like the direction on where to go when finding the ribosome. And so in our pre-mRNA strand, there's two different types of fragment strands, okay? You have you have exons and then you have introns. Exons, they code for amino acids, right? Like they're good. But introns, they don't code for amino acids. So it will just be a waste for the cell to keep these introns and then further translate the introns, because if they don't code for an amino acid, there's no use, because it's not going to make a protein, and that's just going to waste energy. And our cells need to keep as much energy as it can. So it's going to get rid of these introns. How? Well, there's like a certain complex thing, you don't have to know it, that comes, like enzymes that come and they actually just cut out the introns. They remove these introns from the pre-mRNA. And then the new mRNA, which is the mature mRNA, is going to be made by adding adding continuously all of the exons from here. And then they got rid of the introns. And we still have the G cap and the poly A tail. So this is known as a mature mRNA. And then this will now go to the ribosomes in here. RNA splicing occurs like right here. Then it's going to go to the ribosomes, and then we'll leave through the nuclear pores, and then we'll see translation occur. Yeah, thanks so much for watching. Bye.